All right, we're going to be looking at some pretty important concepts today in terms of how this thing is functioning. And by this thing, I mean the heart. We're going to be looking at how the electricity travels through the heart. We've kind of already hinted at it. Now we're going to see how it actually does it. We're looking at the various different parts and pieces. When it comes to heart contraction, there's almost two different separate entities that both are functioning together. We have electricity being initiated, specifically in all of those nodal cells that we talked about, SA node, AV bundle branches, Purkinje fibers. And then we have what the actual heart is doing, the cardiac muscle itself. So we're going to be talking about two separate entities collectively here. So we'll be talking about how electricity is propagating through the nodal cells, and then we'll look at what happens when that electricity gets into the cardiac muscle itself. And shockingly, it's going to be a lot of similarities between us, cardiac muscle, and skeletal muscle, but there are some differences. We looked at the anatomy a little bit, and I don't know if it was abundantly clear, and I was trying to think back, and maybe it's not. So I'll point out a couple things that hopefully is more clear now after I mention it. So let me go on here. All right, so here are the two things that I was talking about. We have the actual conduction system. So we have the nodal cells, okay, and that's what's up here, all right? All those nodal cells, SA node, AV bundle, left and right bundle branches, Purkinje fibers. And then we have what actually happens in the cardiac muscle itself. And that's down here, okay? So we have to have both of these processes working together. We have to have them coordinating this electrical movement through the tissue, nodal cells, cardiac muscle, in a succinct pattern. And if for some reason I cannot get the electricity to move properly from one nodal cell to the next, SA node to AV bundle, then the heart's going to start misfiring, and then that's going to affect how much blood gets pumped out at any given heart contraction. That will affect oxygen within the blood, or oxygen within the tissues, I guess I should say, oxygen to the brain. And that's going to affect the various different receptors and cause the heart to maybe work harder and work faster. And that's not going to be any good either, because we're just going to be still misfiring and not sending enough. And then eventually your body's going to do a hard reset. And by hard reset, it's going to make you pass out. So let's look at what is happening with the nodal cells first. Okay? And we are going to use a bunch of terminology that we've already seen before when we looked at skeletal muscles. Okay, so there's really not going to be a whole hell of a lot. I mean, do these things look different? Nope. At least I hope to God not. Sodium potassium pumps to get everything back to normal. There's some calcium pumps. We've had calcium pumps all over the dang place. Leak channels to maintain resting membrane potential. We still have sodium. We still have, sodium voltage gated channels. We still have potassium voltage gated channels. Okay, so we're going to have those specific ones there. But when it comes to potassium, I should have put it up there in the common. I mean, it's voltage gated potassium channels. I mean, yeah, we've had those before. But there are a couple specific things that are different, and it's the specifics that some of you sometimes get caught up on. And it's that specificity that I want to make sure that you pay attention to. Because there are some differences when it comes to cardiac muscle contraction compared to skeletal muscle contraction. And what I have underlined there is one of the biggies. We have what are called slow voltage gated sodium channels. And we have what's called fast gated calcium channels. And we'll see what they do here in a second, but I bet you can figure it out. Calcium gated channels are going to bring in calcium. Sodium gated channels involve sodium. One is going to be more slower to close, and one's going to be really fast to open. And I imagine by their names, how I just described them, you could figure that out. The resting membrane potential here is about minus 60 millivolts. It's a little bit closer to 0. And hopefully that should automatically tell you that it's going to be a little bit easier to get to threshold and fire faster because of that sheer fact. Okay. And here's just a lovely little picture here. And here's just a lovely little picture here that looks at, that sinoatrial node, the nodal cells. Could be an atrioventricular node, whatever. And there you can see slow voltage gated sodium channels, fast gated calcium channels, and ho-hum voltage gated potassium. Okay. So let's look at what happens with these nodal cells. And the thing to remember, guys, these are autorhythmic. Okay. These cells are spontaneous. They set the pace. Sinoatrial node is the pacemaker cell. It's the pacemaker node. It sets the pace for how fast the heart is going to contract. It gets influenced by the autonomic nervous system, by those things that we talked about, sympathetic and parasympathetic. And when we talk about how we can affect cardiac output in the last little bit of this lecture, we'll be talking about those things. But again, it's going to be a review. You already know it. We discussed it when we looked at the autonomic nervous system and how sympathetic and parasympathetic generally functions. So you already have a basic understanding of what in the hell's going on. So there's not going to be a whole lot of new stuff there for you guys. Nothing. So how does a nodal cell reach threshold? Well, like anything does, it involves sodium, but specifically it's slow voltage gated sodium channels. Again, we are at minus 60 millivolts here for our RMP, our resting membrane potential, and we get to threshold pretty dang quickly, which again is fantastic because, I want this to reach threshold quickly to fire the impulse. Then the nodal cell undergoes a depolarization event. As soon as we get to that minus 40 millivolts, fast voltage gated calcium channels open. They open quickly, hence fast, and calcium comes rushing in. I'll mention it now, and I'll mention it again here a little bit later as well. Heart, cardiac muscle, does not, NOT, have an extensive sarcoplasmic reticulum. It does not have any cisternate, really. It does not have a huge reservoir to store calcium, negligible. Very little bit gets stored inside. Cardiac muscle is going to be dependent upon, the heart is going to be dependent upon calcium on the outside being pumped in. That's why we see voltage-gated calcium channels here with the heart. And we didn't see those necessarily with muscle. Skeletal muscle, sorry. Okay. So as soon as we get to minus 40 millivolts, the slow voltage-gated channels are open and they're slow to close. Okay. Depolarization, as soon as we get to that minus 40 millivolts here, which is technically threshold for the nodal cells here, it goes the rest of the way because of the calcium. As the nodal cell fires, depolarizes, calcium channels close, and just like before, potassium helps to reestablish. Okay. Sodium rushing in and potassium rushing out, ain't nothing new. Okay, ain't nothing new. We're just using the word slow voltage gated sodium channel. The only thing that's added here, guys, is Mr. Calcium. That's it. Okay. That is it. The only thing different. And this is looking at specifically the nodal cells, whether it's SA node, AV bundle. All of them undergo this right here. Slow voltage gated sodium channels open. Sodium comes rushing in. Go from -60 to -40. We've reached threshold. It's going to fire. Fast voltage gated calcium channels open. That takes us the rest of the way. Then we repolarize. As soon as calcium voltage gated channels, voltage gated calcium channels open, slow. Sodium channels start to close slowly. Get our depolarization event, and calcium voltage gated channel quickly closes. And then potassium does what it does. It helps to reestablish resting membrane potential. So really, I mean, nothing really weird and obscure here. SA node is the dude. SA node is the one that sets the pace. It is the pacemaker cell. And you can see about every 0.8 seconds, I mean, it's starting another one, and another one, and another one. If the SA node was the only nodal cell there, this thing would fire a lot more. We have a couple different things. We have a couple different structural differences that exist between some nodal cells that we'll briefly talk about that help to limit how fast the heart can contract. Okay. We talked about limiting factors when we looked at how nerve impulses travel in the nervous system. Okay, they're going down 200 miles an hour down an axon. Good God Almighty, that's really fast. Stuff would move faster if we didn't have this thing called neurotransmitters. Gotta get the neurotransmitters to be released, diffuse across, bind to receptors, kickstart the process again. Okay, even though that doesn't take a whole heck of a lot of time, fractions of a second, it is still slower than that actual impulse moving down the axon. So that movement involving neurotransmitters is the limiting factor. That slows down impulses moving through the nervous system just a little bit. We're going to see some stuff happen here with some of the structure associated with the heart, with the nodal cells specifically, that is going to slow down the heart so it doesn't race to 100 beats per minute or a little bit more. Okay. And you really only see this in a culture where the SA node runs amok and fires way fast when it's just by itself. Okay. You would only see this if you had loss of the atrioventricular node and the atrioventricular bundle. If that's the case, you got other stuff to worry about in your life than your heart racing a little bit. You're having massive destruction of heart tissue. It is this tit for tat, this tit for tat that we see with the sympathetic and parasympathetic nervous system. It's going to be one of the things that affects cardiac output. I know, shocking, right? We just talked about it. Shockingly, that we'd talk about it again here. All right. So there is involvement of the autonomic nervous system to help limit this already. And that's what I talk about here with vagal tone. Vagal tone is just the vagus nerve, right? Cranial nerve 10. It is that parasympathetic input from that vagus that is going to help slow that heart rate. Okay. And again, this is because of that dual innervation going into pretty much every single organ that you have. So here you see my minus 60, my resting membrane potential. Slow voltage gated sodium channels open up. Sodium rushes in. As soon as we get to threshold, minus 40. As soon as we get to that threshold of minus 40, fast voltage gated calcium channels open up. These slow sodium voltage gated channels close. Get an influx of calcium that's going to cause this nodal cell to depolarize. As soon as it reaches just slightly above zero, fast voltage gated calcium channels close. Potassium opens up. And we start to return to resting. And you can see here with nodal cells, I mean, there's not a whole heck of a lot of dipping there, so there's not necessarily a lot of sodium potassium pumps, sorry, brain fart, involved here with the nodal cells. Once it gets to just a smidge below minus 60, it shuts, and then it's ready to go again, okay? As soon as it gets to minus 60, potassium or the sodium immediately opens back up. Potassium starts to close, and we have our pacemaker potential, and it's that influx of sodium that happens again, and we do the whole thing over again, and it happens every 0.8 tenths of a second. As soon as that electricity, as soon as that depolarization event occurs, it travels through the internodal pathway, okay? This is where it starts. Most people forget where it starts for whatever reason on an exam. Sometimes if it's asked short answer wise, they forget, or whether it's in a multiple choice type format that puts them in the correct order, they sometimes forget the first step. And it all again begins up here at the sinoatrial node. From the sinoatrial node, we go through this internodal pathway, and we reach the atrioventricular node. Remember, SA node's in the very top, AV node's in the very bottom of that right atrium. And I mean, it travels through gap junctions. I'm just trying to, weave in some of the words and concepts that we've talked about already before. So these gap junctions are what allow that electricity to quickly move from one cell to the next in the cardiac muscle. That's where we find them a lot, that and some smooth. And it's these gap junctions that allow that electricity to quickly spread from one adjacent cell to the next, and we can get the atria to contract. At the atrioventricular node, this impulse is going to be delayed. Okay. This is awesome. The nodal cells are much smaller, and they have fewer gap junctions. So because they are smaller in diameter, that's going to slow down how the electricity moves it, right? And we saw that with big, thick nerves or very small nerves, right? With smaller diameter and fewer gap junctions, the electricity doesn't move through as quick. And again, I know we're talking like fractions and fractions of seconds here, but it is a little bit slower. This is important. This is important. It allows these ventricles to completely fill before they contract. Would everybody agree that the atria are smaller than the ventricles? Yes. I hope you're all shaking your head yes. Okay. If the atria contracted, pushed that blood into the ventricles, and then immediately, as soon as it contracted, it relaxed it and then contract. If that ventricle wasn't all the way filled, what's the point? What is the point of having that big chamber if I can't fill it up optimally? So this small little delay that occurs at the AV node because of its smaller structure, fewer gap junctions, allows that ventricle to completely fill prior to contract. Okay. It's what allows such a bigger volume of blood within it so it can push a little bit more out. Otherwise, the heart would have to work a lot harder. And if there is a screw up, that's generally what happens. From the AV node, we relay that impulse to the AV bundle, the atrioventricular bundle, old school name bundle of His. We go down the left and right bundle branches, down that interventricular septum. At the apex, the bundle branches stop and it comes back as Purkinje fibers. Oh man, my wrist. As Purkinje fibers. And those Purkinje fibers whoosh, penetrate into the cardio muscle, okay, the myocardium. And it allows that electricity now to penetrate and spread within the cardiac muscle. Again, gap junctions are there, are existing there as well. Okay. And so even though there's a fraction of a second delay there at the AV node, it still allows the heart to contract as a unit, completely contract, starts to relax, and then fraction of a second, contracts those ventricles together. And it ejects the blood through the various different trunks, through the aortic trunk, through the pulmonary trunk. So here's my sinoatrial node, right? In the upper portion of that right atrium. Spreads across the atria through that internodal pathway. Gets down here to the atrioventricular node, slows it down just a smidge. Okay. Slows it down just a smidge, allows this impulse to travel across, travel completely down. Allows these atria to completely contract. As we get from the AV node to the AV bundle, just that slight little delay that occurs there allows these ventricles to completely fill with blood. As the bundle branches take that electricity down and at the apex become Purkinje fibers, Purkinje fibers penetrating that cardiac muscle, I take that electricity into the cardiac muscle. And now the ventricles contract and they eject the blood. SA node, AV bundle, left-right bundle branches, Purkinje fibers, cardio muscle. Where the AV node is a little bit smaller, where the AV node has fewer gap junctions to give us that little bit of delay. Purkinje fibers are very large in diameter, and that allows the action potential to spread very rapidly. Again, it's spreading rapidly to get into all that myocardium within the two ventricles. And again, it's ensuring that these ventricles can contract simultaneously. Again, both atria contract at the same time. Both ventricles contract at the same time. If they don't, the heart ain't working. The heart ain't working properly. Okay? And that's going to screw you all up. Papillary muscle contract almost immediately. And this seems a little weird, to be honest. But the ventricles generate so much force that if the papillary muscle, because remember the papillary muscle are what hold on to the chordae tendineae, and the chordae tendineae are what attach into the cusp of the valves, those atrioventricular valves. If the papillary muscles don't contract just a little bit before the ventricles do, they won't be ready. They need to be ready because they need to exert force on those chordae tendineae to pull down. So when all that force from the ventricles contracting force that blood up, because remember where the semilunar valves, they're up, okay? So as soon as that ventricle contracts and it generates all that force, it's going to force that blood up against those atrioventricular valves. If those papillary muscles aren't just already contracted and ready for all that pressure that's going to get exerted against that cusp, well, when that ventricle contracts, that blood's going to be ejected easily by those cusps. And obviously that's not conducive for a healthy heart. So the papillary muscles stimulate almost immediately as soon as that comes in, right before the ventricle, just again, fractions and fractions and fractions of a second right before the ventricle contracts. Prevents those cusps from being insufficient, non-functional.